Lesson 3.7, the Metabolic Mystery Patient Lab. The goals of this lesson are to give students a hands-on lab experience and to apply what students know about diabetes to a clinical setting by following a patient through her diagnosis. At the end of this lesson, students should be able to explain the primary cause of abnormal blood glucose concentrations, too little or too much insulin. We'll achieve these goals by having students complete a laboratory activity. To prepare for this lesson, you will need to review the key scientific concepts that will be presented throughout the lesson. These include type 1 and type 2 diabetes and the mechanism of insulin on the muscles and blood glucose. You can review this scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure, including instructions on how to manage the discussion and the activity. The student workbook provides additional explanations for students. You will need to print out one activity worksheet per student. This lesson also requires the following additional materials: glucose testing strips, which can be purchased from a drugstore or online, dextrose or glucose powder, which can be purchased by a chemical supplier, or dextrose can be found in a nutrition supplement store, vials or small cups. This lab can also be conducted using pH indicator strips and using acids and bases to make several solutions at a different pH. These would replace the glucose testing strips and the dextrose or glucose powder. The key points of this lesson's do now are to engage students in today's lab by introducing the patient and to go over some symptoms of diabetes, including excessive thirst, fatigue, and weight loss. We'll get there by having students read the overview of the patient on their worksheet. You will want to have students pick out the key information about this patient, like her age, BMI, and symptoms. The key points of the lesson's activity are that blood glucose concentrations change throughout the day depending on meals and exercise. Insulin prevents blood glucose from getting dangerously high, and type 1 diabetes patients are treated with insulin because their insulin receptors are working. We'll get there by having students complete a lab in 2 parts. This lab entails students testing several fake blood glucose samples, which are simply glucose dissolved in water. If you are going to complete this lab as written, you will need to prepare 5 solutions of varying concentrations of glucose before class. Caution, it can take some practice to get these glucose solutions to yield the same colors on the glucose testing strip as we show in the slide, so be sure to give yourself adequate time to prepare for the lab. Alternatively, you can make up your own solution concentrations and adjust the scale you give your students accordingly. The activity worksheet asks students to take notes on those key points, which are data collected during the experiment, comparison of each student or groups of students' data to the class's data, including averages and standard deviation, plotting of data on a graph, explanation of patterns in data, and disease diagnosis and treatment. You can review all of this information in the worksheet answer key. It has everything organized just as your students will be organizing it. Additionally, the teacher manual goes over all of the key concepts. You can also use the worksheet answer key and the teacher manual as a guide to prepare for this lab. This lab is divided into 2 parts. In part 1, students will measure a patient's blood glucose concentration throughout the day using glucose testing strips. Students will determine the concentration of blood glucose in the samples using the scale provided, then compare those concentrations to the table on slide 3. Because you will have several student groups, you may get a variety of results, even though all students are testing the same samples. This is a great opportunity to have a discussion about the variability of results in research, which is dealt with by scientists by running certain statistical procedures to control for that variability. If appropriate in your classroom, you can use this as an opportunity to have students calculate averages and standard deviations using the class's data. The formula for standard deviation is shown on slide 6. At the end of Part 1 of the lab, students will compare the patient's blood glucose concentrations to the typical concentrations from a healthy person. Using this, students should speculate what is causing the changes in blood glucose throughout the day. Before moving on to Part 2 of the lab, have students form a diagnosis for the patient. Students are usually quick to decide that the patient has diabetes, but they won't be able to decipher whether it is type 1 or type 2 diabetes from glucose concentrations alone. You will want to ask the students what tests they could do to determine what type of diabetes the patient has. This alludes to the key point that type 1 diabetics will have low blood insulin because the pancreas has stopped producing it, and type 2 diabetics will have high blood insulin because it is the insulin receptor that is malfunctioning. In Part 2 of the lab, students will again analyze blood glucose concentrations from the patient, but this time the patient has been diagnosed with type 1 diabetes and has been using insulin to treat her disease. This part of the lab follows the same pattern as the first part, but students should find that the blood glucose concentrations dip very low at one point in the day. This dip is caused by the patient giving too much insulin or by the patient exercising without correcting her insulin needs. After the students have completed the lab, you can wrap up the lesson by having students explain what caused the changes in blood glucose concentrations throughout the day after the patient was treating her diabetes. If you would like to shorten this lab, you can divide students into two groups and have one group complete Part 1 and one group complete Part 2 of the lab. Students may ask about the treatment of type 2 diabetes. Because type 2 diabetics can still make insulin, it wouldn't make sense to give them extra insulin as the treatment. Instead, type 2 diabetics are prescribed drugs that help the insulin receptor to function better, and drugs that help lower blood glucose levels by causing the kidneys to flush it out in the urine. At the end of this lesson, students should be able to explain the primary cause of abnormal blood glucose concentrations, too little or too much insulin. Students should have already learned about the mechanisms of insulin in lessons 2.3, 2.4, 2.5, and 2.6. Don't forget that if you have any questions, concerns, or feedback, you can let us know. You can contact any of the CTSE team members and we will be happy to help.